Hello everyone, I'm Dr. Sonia Angelin, Associate Professor, Department of Life Sciences, Christu Jayanti College. In this video, we will be discussing about the enzymology of replication. Everyone knows that replication is very important for all the living organisms, as it is a fundamental property of living organisms is their ability to reproduce. The process occurs by which a single parental cell divides to produce two identical daughter cells. In this process, the genetic material must be duplicated prior to the cell division so that the daughter cells receives a full complement of the genetic information. Replication means synthesis of daughter nucleic acid molecules identical to the parental nucleic acids. Thus, accurate and complete replication of DNA is essential to the ability of a cell and organism to reproduce. DNA is made up of double helix of two complementary strands. During replications, these strands are separated, and there are many enzymes that are involved in the replication process. We will be seeing about these replication enzymes in the following slides. The enzymes involved in DNA replication are DNA helicase, DNA polymerase, DNA clamp, single-stranded binding proteins, topoisomerase, DNA ligase, and primase enzyme. To discuss in detail about these enzymes, firstly, DNA helicase was discovered in E. coli in the year 1976 and are a class of enzymes vital to all living organisms. The pictorial representation also shows the role of helicase, where it is a destabilizing enzyme that separates the two strands of DNA at the replication fork behind the topoisomerase. They are the motor proteins that move directionally along a nucleic acid's phosphodiester backbone, separating two annealed nucleic acid strands. It can be either DNA-RNA or DNA-DNA and RNA-DNA hybrid using energy that is ATP. The next enzyme is DNA polymerase, where this enzyme synthesizes the DNA molecules from deoxyribonucleotides, the building blocks of DNA. During this process, DNA polymerase reads the existing DNA strands to create two new strands that match the existing ones. Also performs proofreading and error correction. These are the two types of DNA polymerases which are existing in the prokaryotic and eukaryotic, where prokaryotic DNA polymerases, there are three different types, polymerase 1, 2, and 3, which has their special activities inside the cell. And the eukaryotic DNA polymerases are 5 in types, and they are alpha, beta, gamma, epsilon, and delta DNA polymerases, which have their own activity inside the cell. The DNA clamp is the next enzyme, where it is also known as a sliding clamp. It is a protein fold that serves as a processivity promoting factor in DNA replication. As a critical component of the DNA polymerase 3 holoenzyme, the clamp protein binds the DNA polymerase and prevents this enzyme from dissociating from the template DNA strand. The single-stranded binding protein is the next protein for discussion, where it prevents the re-annealing of and protects the single-stranded DNA. It prevents the re-annealing of the DNA and also protects the single-stranded DNA from being digested by the nucleases and prevents the secondary structure formation until the process of replication is completed. The topoisomerase is the next enzyme, where every cell has enzymes that increase or decrease the extent of DNA unwinding, and this role is done by topoisomerase. It is also known as DNA gyrase, which act on the topology of the DNA. Topoisomerases bind to double-stranded DNA and cut the phosphate backbone of either one or both the DNA strands. This intermediate break allows the DNA to be untangled or unwound and till the end of the process. The backbone of the DNA is resealed again. DNA ligase is an important enzyme, and it's a specific type of enzyme which facilitates the joining of the DNA strands together by catalyzing the formation of a phosphodiester bond. DNA ligase is used in both DNA repair and replication. The mechanism of DNA ligase is to form two covalent phosphodiester bonds between the 3' hydroxyl ends of one nucleotide and with the 5' phosphate end of another. The other enzyme involved in replication is primase. Primase catalyzes the synthesis of a short RNA segment which is called as a primer complementary to the single-stranded DNA template. Primase is a key importance in DNA replication because no known replicative DNA polymerases can initiate the synthesis of DNA strand without an initial RNA or DNA primer. After this elongation, the RNA piece is removed by the 5'3' exonuclease activity and refilled with DNA. This is a pictorial representation showing the role of each and every enzyme, starting from topoisomerase, unwinding of DNA, helicase, further continuing the unwinding of DNA at the fork, and addition of the DNA polymerase enzyme which adds the DNT piece, and the role of single-stranded binding proteins which enables the DNA to remain as a single strand, and the role of ligase where the DNA joining and resealing is done. Thank you.